Afrotheria is a clade of mammals, the living members of which belong to groups that are either currently living in Africa or of African origin: golden moles, sengis, tenrecs, aardvarks, hyraxes, elephants, sea cows, and the extinct Desmostylia. They share few anatomical features, but many are partly or entirely African in their distribution. This probably reflects the fact that Africa was an island continent through the early Cenozoic. Because the continent was isolated by water, Laurasian groups such as insectivores, rabbits, and ungulates could not become established. Instead, the niches occupied by those groups were filled by tenrecs, hyraxes and elephants that evolved from the ancestral afrothere. This adaptive radiation may have occurred in response to the Cretaceous or Euro-Paleogene mass extinction. The common ancestry of these animals was not recognized until the late 1990s. Historically, the pain ungulata had been linked to other ungulates. The golden mole, tenrecs, and elephant shrews were the traditional insectivora, and the aardvarks with the pangolins and the Cenozoics within the invalid taxon Edentata. Continuing work on the molecular and morphological diversity of Afrotherian mammals has provided ever-increasing support for their common ancestry. Evolutionary relationships. The Afrotherian clade was originally proposed in 1998 based on analyses of DNA sequence data. However, previous studies had hinted at the close interrelationships among subsets of endemic African mammals. Some of these studies date to the 1920s. There were also sporadic papers in the 1980s and 1990s. The core of the Afrotheria consists of the pain ungulata, that is, elephants, sea cows, and hyraxes, a group with a long history among comparative anatomists. Hence, while DNA sequence data have proven essential to infer the existence of the Afrotheria as a whole, and while the Afroinsectivia were not recognized as part of Afrotheria without DNA data, some precedent is found in the comparative anatomical literature for the idea that at least part of this group forms a clade. The Paleocene genus Ossipia, which is the most completely known Paleocene African mammal and the oldest Afrotherian known from a complete skull, shares similarities with both pain ungulata and Afroinsectivia, and may help to characterize the ancestral body type of Afrotherians. Since the 1990s, increasing molecular and anatomical data have been applied to the classification of animals. Both types of data support the idea that Afrotherian mammals are descended from a single common ancestor to the exclusion of other mammals. On the anatomical side, features shared by most, if not all, afrotheres include high vertebral counts, aspects of placental membrane formation, the shape of the ankle bones, and the relatively late eruption of the permanent dentition. The snout is unusually long and mobile in several Afrotherian species. Studies of genomic data, including millions of aligned nucleotides sampled for a growing number of placental mammals, also support Afrotheria as a clade. Afrotheria is now recognized as one of the four major groups within the Eutheria. Relations within the four cohorts, Afrotheria, Cenozoic, Laurasian, and Odontoglyas, and the identity of the placental root, remain somewhat controversial. Afrotheria as a clade has usually been discussed without a Linnaean rank, but has been assigned the rank of cohort, magnorder, and superorder. One reconstruction, which applies the molecular clock, proposes that the oldest split occurred between Afrotheria and the other three some 105 million years ago in the mid-Cretaceous, when the African continent was separated from other major land masses. This idea is consistent with the fossil record of Cenozoic, which is restricted to South America. However, Afrotheria itself does not have a fossil record restricted to Africa, although this does seem to be true for the oldest, undisputed Afrotherians. Furthermore, the correspondence of Afrotherian origins with the Africa-South America tectonic split is not consistent with other applications of the molecular clock or with the mammalian fossil record. More recent, genomic scale phylogenies favor the hypothesis that Afrotheria and Cenozoic comprise sister taxa at the base of the placental mammal radiation. Relations between the various Afrotherian orders are still being studied. On the basis of molecular studies, elephants and manatees appear to be related, and likewise elephant shrews and aardvarks. These findings are compatible with the work of earlier anatomists. Current status and distribution: many members of Afrotheria appear to have a high risk of extinction. Species loss within this group would therefore comprise a particularly devastating loss of genetic and evolutionary diversity. The IUCN Afrotheria Specialist Group notes that Afrotheria, as currently reconstructed, includes nearly a third of all mammalian orders currently found in Africa and Madagascar, but only 75 of more than 1,200 mammalian species in those areas. While most extant species assigned to Afrotheria live in Africa, some occur elsewhere. Many of these are also endangered. Prior to the Quaternary extinction event, proboscideans were present on every continent of the world except Australia and Antarctica. Hyraxes lived in much of Eurasia as recently as the end of the Pliocene. The extinct Afrotherian orders of embrithopods and Desmostylians were also once widely distributed. Organization: Afrotheria is a clade of placental mammals, the STEM designation for which is Eutheria. Based on precedent, some clades are junior synonyms and arguably should be replaced. Afrotheria, a Euroocipia, a Euroiopsodontidae, clade Insectivores, order Macrocelidae, elephant shrews, order Afrocericidae, tenks and golden moles, order Tubulidentata, aardvark, order Eurobibimologasia, order Eurotolmeidae, poorly understood carnivorous mammals. Order Euroafredentata. Clade Painungulata, order Hyracoidae, Hyraxes or Daces, order Proboscidae, elephants, order Sirenia, dugong and manatees, order Desmostylia, order Embrithopoda. See also, Evolution of mammals, Fauna of Africa, Notes. References: Kriegs, Jan L.A., Gennady Churkov, Martin Kiffman, Ursula Jordan, Jürgen Brosius, Jürgen Schmitz. Retroposed Elements as Archives for the Evolutionary History of Placental Mammals. Ploes Bile 4, E91 doi, 10.1371/Journal PBIO. 004091. PMCA 1395351. PMIDA 16515367. William J. Murphy, Eduardo Isarique, Mark S. Springer et al. Resolution of the early placental mammal radiation using Bayesian phylogenetics. Science 294, 2348-2351. doi, 10.1126/Science 1067179. PMIDA 11743200. Seifert, Eric. Gillen, J.M. A New Estimate of Afrotherian Phylogeny based on simultaneous analysis of genomic, morphological, and fossil evidence. BMC Evolutionary Biology 7, 13 doi, 10.186/1471-2148-7-224. PMCA 2248600. PMIDA 17999766. External links: IUCN Species Survival Commission, Afrotheria Specialist Group. Information on the members of Afrotheria, with pictures. Evolution of the mammalian placenta revealed by phylogenetic analysis.